Hello everyone, I'm Dr. Arpita. A very warm welcome to my plant physiology class. The topic for today's discussion is Phytochrome, Cryptochrome and Phototropin. We are going to discuss these topics in brief. I'll only emphasize on the most important points here and would request you to jot them down and practice questions which are related to this topic. In case if you get stuck anywhere, feel free to get back to me in the comment section. Okay, so let's start. So we'll start with phytochromes. Okay, see, these phytochromes are soluble proteins, okay, that exist as homodimers. If you take a look at this diagram, you will be able to tally it with the write-up. See, it has got two identical subunits. Homo means same. Two identical subunits or homodimers, which has got a covalently attached chromophore group. Okay. The first domain is associated with photoreception, or it is called photoreceptor activity, and the second domain is for kinase activity. All right, the second domain is for kinase activity. Clear? The molecular mass of the apoprotein monomer is approximately 125 kilodaltons. Okay. They are synthesized in the cytosol. Now, this is an important point. Please note it down. Where are they synthesized? They're synthesized in the cytosol. Okay. And they assemble autocatalytically with a linear tetrapyrrole chromophore, which is the phytochromobulin. Clear? See, now we'll just write a bit. Okay. So what did I say? What did we just now discuss? These phytochromes are photomorphogenic pigments. What are they? These are photomorphogenic pigments. Here, these are photomorphogenic pigments containing a chromophore. Containing a chromophore. Clear? They contain a chromophore, which is known as what is that known as? Hmm? Which is known as phytochromobulin. Correct? Known as phyto. Clear? These phytochrome is encoded by pi gene family. This one. Look here. It is encoded by the phytochrome gene family or pi gene family. Okay, there are two types to it. Pi A belongs to type 1 category, and phi B, phi C, phi D, and phi E belongs to type 2 category. Clear? See, if we just make an attempt to draw it, you will understand this better. See, This is the N-terminal. Okay. P's domain. Then we have the GF domain. Then your 5. Then is the hinge region. Okay, this is the hinge region. This is your P. Wait a second. And the C terminus. This is the C terminus. Okay, got it? Now I'll change the color. You'll understand better. This part. This part is the photosensory domain. This is the photosensory domain, and this part is the regulatory domain. Clear? Regulatory domain. Got it? See, these phytochromes, they exist in two forms. Okay, the PR form and the PFR form. The PR form or phytochrome red form absorbs red light approximately 667 nanometers wavelength. The absorption of red light converts PR to PFR, which is the active form of the phytochrome protein. Clear? See, we'll write this. What did I say? We'll write. This is the PR form. We'll use two arrows here. This is the PFR form. Okay. This is your active. This is your active state, and this is the inactive state. Okay. What do we have here? We have red light. That means what? PR, inactive PR form gets converted to active PFR form when it is exposed to red light, and active PFR form gets converted to inactive PR form when it is exposed to far red light. All right. So this is called dark reversion. This is called dark reversion. Dark. Clear? This is called dark reversion, and it happens at night. This dark reversion happens at night, and this one is the day event. It happens during day. Okay. See, PFR or this phytochrome far red form, which absorbs far red light approximately 730 nanometers, and this absorption of far red light converts PFR to PR, which is the inactive state of phytochrome protein, right? The absorption of red and far red light alters the chromophore structure which affects the conformation and activity of the phytochrome protein to which it is attached. Clear? See, these two photointerconvertible forms are collectively known as phytochrome system. So this phytochrome system is composed of this PR form and PFR form. By now you know this PR form is the inactive one and this PFR form is the active one. Okay, all right. Now we'll talk a little about its types. We had just now discussed, if you remember, we had just now discussed its types, right? Type 1 and type 2. Okay, type 1 and type 2. Type 1 includes what? Phi A, and type 2 includes phi B, C, D, E. Okay, now see this phi A. Please note this, I'm going slow so that you don't go wrong at any point. Phi A or this type 1 phytochrome is light sensitive. All right, it is light sensitive. So in light, the transcription of Phi gene is inhibited. Fine. Now mRNA degradation and proteolysis takes place, and type 1 phytochrome mainly function in the dark. Mainly functions in dark. This means what? It is activated by far red light. Clear? Now see, now coming to type 2. Type 2 phytochrome means phi B, C, D, and E. Right? Now these are light stable. These are light stable. So present in both light and dark grown plants. In both grown plants. Okay. The most prominent among these, the most prominent among these is this B. Phi B is prominent in perceiving red light. Perceiving red light. You will have to remember this. Okay. You will 100% not forget this. Clear? Next, see, we'll briefly discuss the mechanism or mode of action of phytochrome. See, this one, COP1, what is that? Constitutive photomorphogenic 1. It is what? It is a E3 ubiquitin ligase. E3 which are involved in targeting proteins like HY5, okay, for 26S proteasome mediated degradation. Fine. It inhibits photomorphogenesis. See, this one you take a note. It inhibits photomorphogenesis in dark. Inhibits clear. It inhibits photomorphogenesis in dark by degrading the PIX, that is phytochrome inducing factors. In dark, COP1 is present in the nucleus. An important point. This is present in nucleus in dark. Right? But when there is light exposure, it comes to cytoplasm. When there is light, you will find this in cytoplasm. Is that clear? This one, find this one. When it is dark, it is in the nucleus, and when there is light, it is in the cytoplasm. Fine. So what happens? See, in dark and in light, what happens? When it is dark, that is, there is no exposure to light, what will happen? This COP1 will be in the nucleus and it functions as E3 ubiquitin ligase, and it targets HY5 for degradation by 26S proteasome, as I told you, correct? And there is no photomorphogenesis. But once it is exposed to light, this comes to the cytoplasm and there is high amount of HY5. Why? Because there is no degradation of it and photomorphogenesis will occur. So this is the basic mode of action of this particular protein or phytochrome. Here, now we'll come to cryptochrome. Cryptochromes are also extremely important. You must know about its functions, all these stuff you should know because these are very important from the point of view of your exams. So what are these? These are photoreceptors that regulate entrainment by light of the circadian clock in plants and animals. They also act as integral parts of the central circadian oscillator in animal brains and as receptors controlling photomorphogenesis in response to blue or ultraviolet lights in plants. Right? So what does it say? As you all know, this light-induced growth of plants is called photomorphogenesis. I'll not have to say all these things. So these little ideas you already have. This photomorphogenesis, what is that? It is the light-induced growth in plants. Correct? Now see, this cryptochrome is a blue light receptor. Why am I using this blue color to write now? So that it clicks into your mind and you remember. So what did I say? Cryptochrome is a blue light receptor chromoprotein, blue light chromoprotein. It's a blue light receptor chromoprotein that is very, very important in perception of blue light in plants and its subsequent growth. Okay. Now see the chromophores that are present are chromophores are present, right? The chromophores that are present, they are flavin and pterin. Flavin and pterin. Here, flavin and pterin. All right. Okay. Now we'll first learn a little about the functions of cryptochrome. Very important. I have seen different types of questions coming from this. Functions of cryptochrome. What are they? Plays an important role in generation and maintenance of circadian rhythm. Generation and maintenance of circadian rhythm. That means what? Circadian rhythm means what? The biological clock. Next, see, it helps in anthocyanin production. It helps in anthocyanin production. Next, it plays an important role in stomatal opening. Hmm. It plays a very, very important role in that. Clear? Next, what does it do? See, these are, I'm not writing this is not a function, but you should know these are evolutionary, old and conserved proteins. Huh? It inhibits stem elongation. It plays an important role in phototropism. Wait, it plays an important role in phototropism, photomorphogenesis, huh? Light capture, all these things. Apart from that, it has got a very important role in inhibition of stem elongation. Inhibition of stem elongation. Correct? Now we'll discuss a little about its structure. This cryptochrome is a dimer protein. See? Okay. This cryptochrome is a dimer protein. I'll just write bullet points here. It's a dimer protein. Now, its end terminal has PHR. PHR means photolysins homology region. N terminal has PHR, which is PHR means what? Photolysins homology region. Photolysins homology region. Okay, it has the photolysis homology region, and PHR has two chromophores. Hmm. This PHR has two chromophores. They are what? Flavin, pterin. I had discussed this earlier, right? Flavin, pterin. Correct? This pterin is actually represented by NTHS, which is methynyl tetrahydrofolate, right? See, dimerization is essential for its biological activity and is maintained by N-terminal domain. This N-terminal domain maintains its dimerization, this one, which is important for its biological activity. The C-terminal domain transduces the light signal by interacting with the E3 ubiquitin ligase, that is COP1. I told you earlier, constituted photomorphogenic 1. Okay. Next, see what are the cryptochrome genes. Cryptochrome genes. What are they? Cry1 and Cry2. Okay, this Cry1 and Cry2 are nuclear genes. Wait, I will make a table type like this. Cry1, it is important for your circadian clock. Cry1 is a nuclear gene and it is extremely important in this circadian rhythm part. Okay, circadian clock. Next, what do you have? Cry1 and 2, they are important for induction of flowering. Right? Cry1 and 3, these are light stable. These are light stable. But remember, Cry2, this Cry2 is light sensitive. Only 2. Cry2 is light sensitive. So Cry1, Cry1 and 2, these are nuclear genes. Huh? I'll change the color, you will not forget. These are nuclear genes. Cry3, Cry3 means chloroplast and mitochondria. Cry3 is chloroplast and mitochondria. Okay. Remember, this Cry2 shows a large increase in blue light stimulated cotyledon expansion. So remember this much for cryptochrome, and the last topic for today is phototropins. Phototropins are also equally important Phototropins are plant-specific blue light receptors. These are plant-specific blue light receptors for phototropism, chloroplast movement, leaf expansion, and stomatal opening. Right now, all these responses are thought to optimize photosynthesis by helping to capture light energy efficiently, reduce photo damage, and acquire carbon dioxide. Fine. So, phototropin, these are also, I can just write this for you here. Flavoprotein flavoprotein, 1 second. Okay, so these are flavoprotein acting as blue light receptor, acting as blue light receptor. Okay, remember this. See, they regulate entrainment by light of the circadian clock in plants and animals, right? They also act as integral parts of the central circadian oscillator in animal brains and as receptors controlling photomorphogenesis in response to blue or ultraviolet light in plants. Fine. See, this they have got they play a very important role in optimizing photosynthetic efficiency. That is what? That is phototropism, light induced stomatal movement, chloroplast movement, all these things. Physiological functions of phototropin are conserved in ferns and mosses. Just know this much. Don't have to get into the details, right? Just know this much. That is enough for you. Two types of phototropins are there. Port 1 and port 2. You can see these here. Hmm? This port 1, port 2. Hmm. They have overlapping as well as unique functions. See, port 1 promotes cotyledon and hypocotyle growth. Let's write. What do they do? Cotyledon and and hypocotyle growth. Now, what is the other one doing? What does it do? Promote chloroplast movement. Very important. Huh? Promote chloroplast movement. Fine. Also, phototropism, stomatal opening, all these are important. Fine. Now, these are its function. Now, just a little bit of structural domain we'll talk about. See, I'll just draw it out for you in a very simple manner. Hope it will help you. This is the N-terminus, right? Next one is serine threonine kinase. Serine threonine kinase. This is the C terminus. Here. Now, this is your FMN, which will be here. This is the photosensory domain. This is the photosensory domain. Now, see, this FMN is the chromophore. Hmm? This FMN is the chromophore, right? Now, this LOV is the light oxygen voltage. Okay. This LOV1 and LOV2 bind the chromophore FMN. You can see from here, this LOV1 and LOV2, they bind the FMN together. Huh? So, no? Now, this blue light irradiation or photoexcitation of proteins bound FMN cause a conformational change of phototropin that triggers autophosphorylation and start the phototropin signaling. Now, in dark, what will happen? FMN binds to LOV with non-covalent bonds, and in light, this will the same will happen, but the bond is covalent. Clear? Now, see, photoexcitation of LOV results in activation of C-terminal kinase domain. This part, see, the C-terminal kinase domain, which leads to autophosphorylation of multiple serine residues. Remember this, fine? Now, just before we end, just a little bit of function we'll talk. Phototropin, it mediates oxygen gradient change of plant in light. That means what? The plant bend towards light. Phototropism, you know that. Now, the dark facing side has higher oxygen concentration, which results in rapid cell division and elongation at the darker side. What happens in phototropism? You know that well. And the most important is the chloroplast movement. Okay. In leaves, in an adaptive feature that occurs in order to control light absorption and permanent photo damage. So just remember at low light intensity and high light intensity, what happens respectively, what type of chloroplast movement happens. So when there is low light, I'll just make a column here, I'll write here low. When there is low light intensity and high light intensity, what happens? Chloroplasts move at the upper and lower surface. The chloroplasts move at the higher and lower surface. And here what happens when there is high light intensity? They move to the surface parallel to incident light. Surface parallel to incident light. This is what happens. This will allow this is to minimize light absorption since it is already high. It needs to minimize light absorption, and in the previous case when it's low, it allows maximum light absorption. So this is all about your phototropin, right? I hope you have understood the context of our lecture today. If there is any confusion, any doubt, please feel free to get back to me. Okay. Till then, have a nice time. Thank you.